Welcome back, guys. So it's been a long time since I was active on YouTube because of some problems and everything. And so, after many days, I'm coming back and I'll be teaching a very simple topic actually. So, this looks very difficult, this is practically very difficult, but actually, it's very simple from the perspective. Okay, so I will be talking about something known as conditional expression of genes. Now, when I'm saying conditional expression of genes, I mean at certain condition a specific gene is expressed or it is transcribed and translated from the DNA sequence. Now, one thing, if I ask you, suppose you want to buy a chocolate, okay? Suppose you have a favorite chocolate of yours, okay? So whenever you need to eat that chocolate, you will go to your shop, nearest shop actually, get the chocolate, or do you take a whole truck of chocolate and dump it in your backyard or something like that and eat whenever you want? Because if you do that, you will get fat and you will die. Please call me at your funeral because I want to eat good foods, and please bring DJ. Okay, leave that out. So conditional excission of a gene, okay, it's not expression, excission of a gene. Suppose I want to study particular things when a gene is expressed and how when I delete that gene after a certain time it affects the organism as a whole. Okay, so these kind of things are basically done on a model organism called Drosophila melanogaster. So it's a fruit fly, nothing else. So in this we actually create a plasmid. Okay, that plasmid contains one heat shock sensitive promoter. Okay, HSPR1 is a heat shock sensitive promoter. Along with that we have this GAL4. GAL4 is actually I'm teaching you GAL4 GAL8 UA system. Okay, and also we have we also have this terminal sequence. I written this as FLP, but it's a different concept, FLP FRT, but it kind of like same. It's kind of like same, so I'm just trying to explain it. Also we have this FRT along with gene of interest, which can be any gene. It can be a gene which makes you fat, it can be a gene which makes you thin, or even it can be a gene which makes you dumb. So this FRT as well as GUI and a GFP, provided that after this GFP we have got a stop sequence, and there is a TER termination sequence I have written along with the FRT, and this is nothing but PSG4. So okay, my leg. So this PSG4 is nothing but it's just promoter. Now this promoter gets active when and only this GAL4 is expressed. Now what is happening is that suppose we are actually there is something called allele and everything is there. Suppose we are taking an organism which is actually negative, totally negative, wholly negative for this gene of interest. We don't find this gene of interest when it is when it is developing into that organism, particular model organism we can say. So in this kind of systems, what I want to do is that I want to know the thing which happens when the gene is expressed at a later stage in the lifetime. So for this kind of experiments we need this heat shock promoter plasmid. Okay, so actually what we do is that we actually transform, or actually we incorporate these both plasmids into the cell. Now one thing you must remember, that this GAL4 is inducing this FRT to recognize the this FRT to be active, and whenever this FRT is active, this gene of interest is transcribed and translated. So this GAL4 is like a timer. Whenever I am inducing some kind of cellular stress, or when I am inducing some kind of other mechanisms for activating this GAL4, it can be anything. It can be a heat shock, because I have said the heat shock one. It can be any other gene. Okay, for example, beta galactosidase gene also we can incorporate, but generally heat shock promoter is used in case of Drosophila melanogaster. Now what's happening is that whenever this heat shock protein is activated, this in turn transcribes the GAL4. Now this GAL4 goes and binds to this FRT, or in case of FLP FRT system, this is replaced by FLP, and this FLP is nothing but this FRT is actually flipase recombination termination sites, FRT. Okay, so this FLP goes and binds to the FRT, and thus this means that FLP FRT will now induce the mRNA to be formed from this segment, this particular segment I am marking. And this gene of interest is transcribed as well and translated along with the GFP. So whenever the cell is actually carrying on this, the presence of this GFP expression helps us to determine that okay, this cell is now active. So we inject this, okay, this gene of interest is expressed. As soon as this gene of interest is expressed, we can now study the phenotypical effects. For example, in flies, many of the molecular biologists, what they do is that flies contain different kind of wing patterns, or even they have different kind of antenna patterns and antenna locations. Suppose I want to introduce the fly's antenna in its bellies. Okay, so a fly contains cell in all of its, throughout its body. Okay, it's not that a fly is a single cellular. If you do this, I will kick your A off. Okay, leave it. Well, I don't know what the meaning of A is. I am very good, ma'am. Okay, leave it. So this thing which is I am talking about is this. Suppose I want to express the antenna not in the proper place but also in the belly after a particular time of its life. Suppose the fly is 2 weeks old. Okay, so from its development, that what I do is that in its very singular form or its very basic form, I induce or inject these both systems in the fly. Now I particularly choose some kind of promoter, tissue specific promoters. For example, if I want to make the fly forgetful, I can use some kind of neuron specific promoters, which actually goes and messes up with the fly's neurons. Okay, neuronal cells actually. In this case, I will take the type of promoters present in the fly's bellies. Okay, and now I take the promoter and I keep it over there. And that's a heat shock sensitive promoter. So whenever I am going to heat shock it or cold shock it, it is going to express. Now from that, the GAL4 is expressed. Now this GAL4 is going and recognizing this GAL80 system. So whenever GAL4, GAL80, because in case of this FRT I have written, it's actually complementary to GAL80, nothing else. So whenever this FLP and GAL80 is in play, what it does is that the this actually is a promoter. Okay, these are all promoters. Whatever I am talking about, GAL4, GAL80, these are all kind of promoters. So nothing is that GAL80, it occupies the US and it does not allow GAL4 to be transcribed. Hence GAL80 is kind of an inhibitor. So GAL80 in this case is an inhibitor. But what we are trying to do is that whenever we are heat shocking it, the GAL4 will now go and bind with GAL80 and makes it inactive. The whole complex is now inactive. Or this FLP FRT system is the similar kind of system where it becomes inactive, and whenever it is getting inactive, what it does is that, so let's actually go stepwise, because otherwise it is going to get very difficult for me to make you understand. Suppose suppose take this example. Okay, let us take this example where we are taking some kind of gene of interest. First one, we are taking some kind of gene of interest, followed by we are taking a promoter, tissue specific promoter. It can be actin promoter, it can be any kind of muscular promoter, neuronal promoter, it can be anything. So suppose we are taking a actin promoter. Okay, because actin is present in all kind of cells, actin promoter is present, so we are taking actin promoters except for neuronal cells. So we are taking this actin promoter. Now what we are doing is that we need some activator sequences, nothing but this promoter sequence actually that I am talking about. Now whenever we are inducing this neuron specific or actin specific promoter along with the GAL4, and also we have got some GAL4 sensitive promoter, there is a FRT I am trying to do, basically PSG4 is nothing but GAL4 sensitive. Okay, so when we are doing this, whenever we are doing this, what we are trying to induce is that we are actually trying to incorporate both of these same plasmids into both of these plasmids in the same cell. Now when we are doing that, this gene of interest is getting transcribed, nothing else. See, these both plasmids are injected when it turns 14 years old, or suppose of 2 weeks old in case of fly. We heat shock it, the GAL4 is expressed. This GAL4 in turn binds with the UAS GAL80 system, and thus it helps in transcription of this whole sequence. So GOI, gene of interest, is expressed. Now the fly will start developing antenna in its bellies. Okay, although this kind of model organisms does not survive for long, but okay, let's take example that we want to know the phenotypical things or mechanisms involved in this kind of systems. So what we do is that this GAL4 and UAS and gene of interest is expressed, and the GFP is positive for this. Now we can also do this in other way, okay, because this system is actually not foolproof. Why not foolproof? Because we are talking about the gal4 binding to gal80, okay, and expressing the gene of interest. Now it may happen so that some of the cells are not properly expressed, and thus they are not a part of the the gal4 gal80 UAS system. So in that case we need something more appropriate and something a coordination in the genetic level, and for that we need this siRNA technique. Nothing but the same thing is carried on, but the heat shock enables the gal4 to activate a particular GOI which is actually complementary to the gene of interest we want to target. Suppose we want to delete a gene, okay, that whenever this gene is expressed, suppose I am going to say this like this. Suppose a human is growing, okay, a child is growing, for example. I want to limit its height at a particular stage of the life, at a particular centimeters or a particular feet or something like that. I want to limit its growth up to that only, and see what kind of phenotypical or what kind of other changes we are experiencing in that subject. So what we do is that we take that individual in its very unicellular, in its very simple cellular state, and now we incorporate both of this into the cell. Now this heat shock sensitive promoter is not active until and unless I am inducing it to be active, so that whenever suppose a child has reached, for example, say 123 centimeters, then I am going to heat shock the child. Not I am going to put the child in a boiling water, but there are actually, apart from heat shock, there are many other techniques. Suppose I am using one of the techniques to activate this promoter. Whenever this promoter is active, it's actually a part of Preloxp system right now, but it's all similar kind of systems, okay. So this gal4, for example, this gal4 is induced. So this gal4 will go and bind to its sequence gal4 recognition sites in the other plasmid, and then it will enable the gene of interest to be transcribed, or in this case the complementary gene of interest to be transcribed, that is the reverse sequence of that. So it's producing a small interfering RNA which is going to the transcribed RNA of the original gene and thus getting dimerized. So suppose if this is my original gene sequence, as we get, our complementary genomic system sequence will be like this, and both of them will actually dimerize, and this dimer formation will now induce some cellular dicer for destruction, and thus the GFP here will not be expressed. Hence we can properly say that we have terminated the gene sequence, and now we can examine or see what changes we get in the organism. This is how this kind of systems are monitored. So it's called conditional excission of a gene or conditional silencing of a gene. Gene silencing concept is this. So after many days I am coming with a very simple video, although I will be providing a different video today only if I have enough net and enough time to upload it. But these are very simple thing, okay, simple excision of a gene technique. Although there are many different kind of other excisions like ectopic excisions and different things, these are actually in theoretical way it's very complex. It's not very that complex, but in practical it's very complex. Expressing a gene and incorporating both of these at the same time is kind of a challenge, but yeah, we can do it, and obviously there are many results which has that, and also there are many flies which are sacrificed for this kind of experiments, but we can now monitor and we can see what kind of other things are there. For example, I'm going to give you a case study. It was from my college slide making. So I was doing an enzyme and its moonlighting function called the galactokinase, where there was something as calcineurin induced cardiomyopathy. So in that, what they did is that they designed a similar kind of system, but they replaced this thing with something known as transposon. And that transposon is activated only at a particular only at a particular point in its time, such that it goes and disrupts the galactokinase gene by homologously recombining with it. And transposon is a different concept, I know, but they are trying to jumble up between them. So I'm not going to use so much of jargons like TNACT K, CANACT K. I am not going to give about those jargons. But the simple thing is that they also induce transposon mediated inhibition of the gene, and thus after that they learned that how galactokinase, the galactokinase 1 is the GALK1 gene, is helping in the cardiomyopathy of the Drosophila melanogaster by inducing heart capillary wall diameter changes and also reduced contractility of the heart. That's a different concept altogether, but this is how this kind of models are used in practical life. It's a part of RDT as well as molecular biology. So it's a good combination. This thing looks a bit complex, but I think I may have tried my level best to make you understand this. And also, this is it about the video. So let's meet at the next part of this video and also next video as for my channel, because I was not uploading for many days. Thanks a lot for watching. Please like, share and do subscribe, because I am trying with a new initiative and also I am not that much potent enough to continue this kind of videos without your support. So I must require that kind of support from your side also. And also, if you are not getting anything, if you have got any kind of questions, please ask me. My DMs are almost always open, and also my Facebook accounts, you all know that where it is and how to reply, how to text me actually using Messenger, obviously, if you are not dumb. Sorry for that. Please don't hate me. And now I will like to conclude my video. Please like, share and subscribe, and do support my channel. That's it. Thank you for now. Bye. Ta-ta.